Okay, thank you very much. Welcome to this seventh edition of the workshop. We are just waiting a couple of minutes and then we will start. Okay, Luisa, do you think maybe we can start? Because there is a presentation, introduction, and at 10:20, the first speaker, which is Francesco Bianchini. Okay, so welcome again to this seventh version of the workshop, Synthetic Biology, Artificial Intelligence, and the title is What Can Synthetic Biology Offer to Artificial Intelligence? Strategies and Perspectives for Embodied Chemical Approach to AI. This is the seventh edition organized by me, Luisa, Damiano, and Yutetsu, Kuruma, and I would let Luisa make an introduction based on this topic. So this will serve to guide the discussion later. Thank you, Luisa.
Thank you very much, Pasquale. Yes, welcome to the seventh edition of our workshop, which I would like to briefly introduce by introducing the programmatic concept around which we build this workshop series that we started to organize with Yutetsu and Pasquale since 2013. Programmatic concept, which is SBAI, Synthetic Biology, Artificial Intelligence, that is a concept that we introduced to support a positive collaboration, a cross-fertilization between synthetic biology and AI, based on what we thought were our two main conditions of possibility of this cross-fertilization, this synergy between synthetic biology and AI. On one side, a twofold historical scientific condition related to the evolution of science, and on the other side, a methodological condition. The first condition is articulated in two scientific events, in two scientific developments that, in our view, are grounding the possibility of an SBAI, that is an artificial intelligence realized through techniques and theories developed in synthetic biology. And the first of these two events is the so-called embodiment turn in cognitive science and AI. That is the emergence between the late 80s and the beginning of the 90s of the last century of a new approach to the study of cognitive structure and processes, to the experimental study of natural structure and processes, cognitive structure and processes, that is new in the sense that is body friendly, poses a positive emphasis on the role of the body in cognitive processes. While the second of this event is the constitution within the field of the sciences of life of synthetic biology as a research domain dedicated to the synthetic study of biological processes, that is the study of biological processes through the construction of chemical models of these processes. And the first of this event can be quickly sketched on the basis of this schematic representation of the evolution of cognitive science and the eye, an hyper-schematic representation, which shows that cognitive science and the eye emerged in the classical form in the middle of the 50s of the last century, after a decade that usually the literature describes as the decade of the cybernetic foundations of cognitive science and the eye. And cognitive science and the eye emerged in their classical form, as I said, that usually is called computationalist. And it's called computationalist because the specificity of classical cognitive science and the eye is that of modeling us as cognitive agents through the model, describing us as cognitive agents through the model of the digital computer. That is the idea at the basis of classical cognitive science and the eye is that cognitively we function as digital computers, in the sense that we receive information from our environment, we process this information, and through this processing we generate internal representation of this environment that within this approach is considered as a sort of independent reality, a relatively independent reality from us. And the correspondence between the internal representation of this environment and the environment itself implies our capability of addressing and solving issues of interaction with this environment. This is an approach that guided research in cognitive science and the eye since the middle of the 50s, when this approach emerged, to the middle of the 80s, when we started to have a growing dissatisfaction among specialists in cognitive science and the eye towards this approach, for many reasons that are linked to theoretical difficulties, but also to the applicative difficulties that then led to the so-called winter of classical computationalist AI. We don't have the time to go in detail about this. The important aspect for us is that this crisis implied the search for alternatives, and at the beginning of the 90s emerged embodied cognitive science and AI as an alternative to classical and computational cognitive science and the eye, where the definition of this new approach to cognitive science and the eye, embodied embodiment, contains a reference to the body, because the idea of embodied cognitive science and the eye is that of addressing the difficulties met by classical or computational cognitive science and the eye by putting a new emphasis, a positive emphasis on the role of the body in cognitive processes. And indeed, this transition in the mainstream of cognitive science and the eye from computationalism to the embodied approach is today under scrutiny with what regard the depth of this transition. As embodied cognitive science and AI is recognized to be a plural domain, articulated in many different research lines, many of which are still describing us as cognitive agents through the model of the digital computer, so maintain aspects of the framework of computationalism. In this sense, many authors today do not see a discontinuity between computationalism and embodied cognitive science and the AI. The difference that is considered differentiating these two approaches, also by the authors that are not recognizing the discontinuity, is a difference related to a different view of the role of the body in cognitive processes, in the sense that, according to the proponents of embodiment, computationalist cognitive science and the eye were still fascinated by the metaphysical thesis of a naked mind, an immaterial computational mind, and on this basis was neglecting or even denying through theses such as the thesis of the multiple realizability of the computationalist mind any role of the body in cognitive processes. While also when they maintain aspects, they rely on aspects of computationalism, the proponents of embodied cognitive science and AI are trying to overcome this approach, to overcome the dualism that they see still at the basis of computationalist cognitive science and AI, and to ascribe a new role, a positive role, a critical role, a crucial role to the body in cognitive processes, which implies also a difference at the level of AI research. While in the context of computationalist artificial intelligence, to model natural cognitive processes meant to build software, to generate software, not hardware, in the context of embodied AI, to model natural cognitive processes means to build complete agents, robots, agents that are endowed with a body, and through their body are interacting with an environment, are perceiving an environment, reacting to an environment, and also through this interaction are solving also cognitive tasks. So this is the first of the conditions, the historical scientific condition supporting SBAI, at least in our view, while the second is the development of synthetic biology as a new area within the sciences of life, which happened at the beginning of the new millennium. And with the positioning of synthetic biology in the sciences of life as a synthetic science of life dedicated to, on one side, the design and the construction of new biological parts, devices, and systems, and on the other side, to the redesign of existing natural biological systems. In particular, synthetic biology positions itself within the sciences of life as, on one side, an engineering science, which focuses also on engineering applications as the production of biological functions or the biological parts to solve applicative issues, but also as a genuine science of life, a life science of the artificial, which focuses on the exploration of biological processes, biological structures through the construction and experimental exploration of artificial models of living systems and living processes, based on the application of a research method that was developed in the context of proto-cybernetics, became the official method of cybernetics, and then the official method of AI with the emergence of AI in its classical computationalist form, which is the understanding by building method, or the synthetic method. Is a method that was already proposed in the 30s by Thomas Ross as a research method involving engineering, design, and production of artifacts in the scientific understanding of natural processes, which with a focalization on biological and cognitive processes. Basically, a method that is proposing to scientists to explore life and cognition, lato senso, through the construction and the experimental exploration of systems that are considered artificial or synthetic because, as Christopher Lanton used to say, are made by man rather than by nature. And systems that, when are able to generate the target natural processes based on scientific hypotheses on how these processes are generated in nature, can be considered models of these processes, because they can be used, they can be considered as means to test the hypotheses that are incorporated in these models. So basically, the application of the synthetic method is articulated in three main steps: incorporating scientific hypotheses on the target processes, on how the target processes are generated in nature and in artificial systems that are able, on the basis of these hypotheses, to produce these processes, then embedding these systems, these artificial systems that, as I said in the terms I defined before, can be considered synthetic models of the target processes in environments that the theories from which we extracted the hypotheses we incorporated in these models are defining as appropriate to generate the target processes, and then comparing the behaviors produced through the interaction with this environment by our artificial systems with the behaviors of the target system, the natural behaviors that are the target of the inquiry, to assess the capability of the theories of reference to generate these processes, where in this process it's very important to study the divergence that many authors consider to be always there between the artificial and the natural behaviors, in order to be able to improve our theories of reference, so have better hypotheses to incorporate in our artificial system and start with a new cycle of application of the synthetic method. Currently, the sciences that are applying the synthetic method are at least scientific robotics, which produces hardware models, that is robotic platforms, as models of biological and cognitive processes, computer science, which produces software models, that is computer simulations of biological and cognitive processes, and also synthetic biology, which produces wet-ware models of chemical artifacts used as models of biological processes and lately also of cognitive processes. And the convergence in the application of the synthetic method generates a symmetry between synthetic biology and AI, which both can be considered as engineering science, because are engineering on one side biological, on the other side cognitive functions and systems, and are both also explorative sciences, in the sense that they synthetically explore through the application of the synthetic method, on one side life, on the other side cognition, by constructing and experimentally studying artificial models of the target processes. So we thought that once science associates, as it does in the context of embodied cognitive science and AI, and its more radical lines, life and cognition, maybe then synthetic biology can be conceived and practiced as a form of AI. And we propose a positive answer to this question, not only considering the developments of embodied AI, the developments of synthetic biology, but also the groundings, the historical background of AI, not only the science fiction groundings of AI, considering that in the theatrical drama Rossum's Universal Robots, Karel Čapek, who introduced the term robots in literature, and then from literature to the scientific discourse, imagine robots as biochemical robots originally. That is, robots that are built by a sort of synthetic biology, are built with synthetic biological materials. But more interestingly, in the context of proto-cybernetics and cybernetics, we had already the reference to wet-ware models of cognitive processes. In particular, for example, in the context of the cybernetic foundation of the synthetic modeling of life and cognition provided by Rosenblatt, Wiener, and Bigelow in their very famous article that opened the year of cybernetics, behavior, purpose, and teleology, which was published in Philosophy of Science in 1943, they propose three theses that are the basis of the programmatic concepts of SBAI. The thesis that is shared by embodied AI today, that biological body plays a critical role in cognitive processes. The thesis according to which then to synthetically model natural cognitive processes, we need to synthetically model the biological body, which embodied AI is doing through software and hardware models, and the thesis on which we are focusing our programmatic concept as BAI is the thesis according to which to be synthetically relevant, synthetic models of the body should not simply reproduce biological behaviors, but should also incorporate theories of the body, the biological body's organization, which, according to Rosenblatt, Wiener, and Bigelow, could be better done through wet-ware models. This was already explicit in the article they wrote in 1943. And where this thesis was supporting the attempt of overcoming the proto-cybernetic standard of the synthetic modeling of life and cognition, that was imitation. The idea was that of overcoming imitation towards and going towards models that are able to incorporate hypotheses on the production of the target behaviors that incorporate theories of the biological organization. And we think that this can be the specific interest of creating an SBAI, of cross-fertilizing synthetic biology and artificial intelligence. The possibility of modeling the biological organization in artificial system, which we think is more likely to be done through wet-ware models, wet-ware artificial systems. So we think that a synthetic biology applied in the field of artificial intelligence could favor the transition from superficial models, merely imitative models of the biological body, that we tend to think that today are the standards of embodied AI, to deeper models or deep models, organizational models of the biological body. Indeed, our idea, based on many debates that we have today in philosophy of AI, philosophy of embodied AI, is that the mainstream of embodied AI is constructing, ideating, constructing robots that are modeling superficial aspects of the biological body, the anatomy, movements, behaviors. There are frontiers lines in embodied AI that still are not producing prototypes, that are targeting instead the modeling of the organization of the biological body. We have, for example, attempts like enactive AI that are attempting of modeling the organization of the biological body in robots through software and hardware models for the moment. And what we are proposing is to do that through wet-ware models. And already with Pasquale, we developed a program based on the theory of autopoiesis, which is autopoietic SB AI. And this is the goal of our workshop, that is promoting research programs that are involving synthetic biology in artificial intelligence research by creating a cross-disciplinary forum locally, but the idea is that of creating a cross-disciplinary scientific community discussing grounding issues, defining possibility limits and ways, and stimulating the development of these programs. This is the 7th edition. We had the first edition in Taormina at ECOL 2014, that second edition at ALife 2014 in New York, ECOL 2015, ECOL 2017, ALife 2018 in Tokyo. Last year, we organized the 6th edition in the online edition of ALife that was organized by a Prague research group. And this year we are proposing this new online version of the workshop. I leave the stage now to Pasquale to present our workshop, and thank you very much for following this brief introduction.
Thank you. Thank you, Luisa. Actually, it was a quite deep introduction with many interesting ideas and important historical reconstruction. We have time at the end of the workshop to make a discussion about these themes. Okay, now we can start. We are just 5 minutes late, but fortunately there is some buffer time to recover. And so I would like to introduce Francesco Bianchini, who is a professor of philosophy at the University of Bologna. He's our first speaker. And so I invite you, hi Francesco, to activate the microphone and share the screen.
Okay. Thank you, Pasquale, and thank you, Luisa and Yutetsu, for admitting me to this 7th edition of the workshop. I share the screen. Okay. And I start the timer, even though I think you can remind me if I'm going too long on time. And so I want to speak today about this long title, the organization from line to cognition and the current approaches in AI. Last time I spoke about the relationship between synthetic biology and artificial intelligence, because it's the core of this workshop. This time I want to speak of something that is related. Especially, I want to focus on the notion of autopoiesis that is very strongly related to the main issues of this workshop, or I think that it is. And I want to stress something that is connected to one of the main features of autopoiesis, that is organization. I want to show how the autopoietic view is related, or we can speak about an autopoietic view in some approaches, current approaches in artificial intelligence. And I will suggest that there is no autopoiesis in the two main corpora of approaches to cognitive artificial intelligence, and I want to suggest what kind of approach is a good candidate to use the autopoiesis features fruitfully to understand the relationship between life and cognition that is very important in autopoiesis, but and also in the relationship between synthetic biology and artificial intelligence and in the wide field of artificial life. But I think there is still a gap. So, okay. So autopoiesis is a 1972 term, or in the 1972 was the publication of the well-known, very well-known book of Maturana and Varela, Autopoiesis and Cognition, the Realization of the Living, in which the notion of autopoiesis and the notion of cognition were posed in a strict relationship. And also this notion is a notion that connects life and cognition. 1972 is before the turn of which Luisa was speaking a few minutes ago, turning cognitive science and artificial intelligence, but it is reasonable to think that and to say that autopoiesis anticipates some notions that, or some ideas that has been very important in the turn to the more contemporary artificial intelligence and cognitive approaches. So autopoiesis refers to systems that are able to maintain and produce themselves by producing their own parts. Maintain and produce, two things. And the main example are cells in the first place, but then it was extended to other kinds of systems. In the 1972 books, we can find this very interesting definition of autopoietic machine. An autopoietic machine is a machine organized. I have underlined the words that are to me very interesting. Defined as unity as a network of processes of production, transformation, and destruction of components, which, one, through their interaction and transformation, continuously regenerate and realize the network of processes, relations that produce them, and two, constitute it, the machine, as a concrete unity in space, in which the components exist by specifying the topological domain of its realization as such a network. So why is so interesting? I think that these are the very main relevant characteristics of organization. The unity in the space, but also unity as autonomy, because they are systems that are able to maintain themselves. And the stress of on the relationship between autopoiesis and organization is also recalled in an article, the first article in which autopoiesis was quoted or introduced in 1974 on biosystems. And in that article, we can find other interesting quotations. So the idea, for example, of an autopoietic organization that is a unity by a network of production of components which, one, participate recursively in the same network of production of components which produce these components, so it's a dynamical process, and to realize the networks. Network is a word that is very recurrent in the definition of autopoiesis, and today is a standard word, standard notion in artificial intelligence, but was not in the 1970 or in the 70s of the last century. Of production as a unit in the space in which the component exists. So the case of the cell is the main one, a network of chemical reactions which produce molecules. And so I think that there is a strong relation between network and organization that the autopoietic view underlines for the first time in a very strong way. Organizational systems from the point of view of autopoiesis are living systems, are also the living system that are subclass of mechanistic system. This is also in the 1974 article. And so we can see also a sort of mechanistic view in that article. We are in the first part of the history of AI, so it's not so strange. We can find an idea of multiple realizability that is typical of the computational view on simulation or cognitive science processes. So the same organization may be realized in different systems with different kinds of components, as long as these components have their properties which realize the required relations. It is obvious that with respect to the organization, such systems are members of the same class, even though with respect of the nature of their component, they may be distinct. So this is a very strong claim that is aligned to contemporary research on synthetic or that use synthetic methods. So organization as the core of this effort. So the stress on organization is undoubtable in this first introduction of autopoiesis, but there is also another interesting thing, that is the requirement that is at the very beginning of the article, that the autopoiesis is not committed with the history-dependent processes. Organization and organizational view is not in history-dependent processes, and autopoiesis as organization is as the basis of life. So we can think that it could be considered also the very basis of ALife. No? We can try to, or I want to try to extend this notion to the consideration of life from an artificial point of view. And I want to suggest that there could be also a strong connection with the relationship between synthetic biology and artificial intelligence. So what is the relation between autopoiesis, organization, and artificial intelligence? We can think that from the cell, that this is the first example of a autopoietic system, we can extend this notion, and this is, has been done to artificial system. And we can also see the fact that autopoiesis, that is strongly connected to the or previous notion, like the homeostatic view and so forth and so on, that the autopoietic living system, or living systems that are autopoietic in nature, could be also an example, something that could be simulated, and we can arrive to create artificial living systems within the view of autopoiesis. But two questions are more open. How can we move from living system to cognition or to cognitive system? It is not very very clear or very neat, this passage to me. And how can we connect autopoiesis to the artificial intelligence that is devoted to build modeling cognition? I think that we can go to consider from this point of view the different AI approaches. There are today many different approaches in artificial intelligence to different parts of the simulation of intelligence or cognitive processes or whatever. I just have given a little list because the approaches in artificial intelligence depends on the notion of intelligence, the available technologies that are always changing, the notion of system, the notion of autonomy, the aims of artificial intelligence, the different intelligence capabilities. We want to model language, reasoning, creativity, sensory motor abilities, and so forth and so on. The role of body, the role of brain, and at the end of the day, especially the notion of cognition. I chose two present-day approaches that to me are relevant in this to analyze the possibility today of the notion of autopoiesis in connection with artificial intelligence. My claim, my hypothesis is that if you consider autopoiesis and artificial intelligence, and if autopoiesis is focused on organization and not on history-dependent processes, especially evolution and reproduction, organizational artificial intelligence system specifically related to cognition should encompass autopoietic features or mechanisms. So I want to stress, to underline that autopoiesis is not a notion of the past and could be useful, but we have to see how. So to me, this is true, and I want to show how this perspective is fruitful. If we consider the first approach, the approach of cognitive architectures, that is typical since the 60s or 70s, still until today, the approach that is exemplified by cognitive architectures like SOAR, ACTR, and also other, and that is we can consider it in within the idea that we can have a cognitive design. We can design cognitive models. We may say that a lot of these models use a notion of organization, very strong notion of organization that is related to a sort of modules, network integration, and information exchange. That is, in this cognitive architecture, we can find cognitive models, computational models, so because they are models, computational models in simulation. That use symbolic information that are inspired to a cognitive general models. But this approach, that was the one that in the history of artificial intelligence and cognitive science is the one that is more closer to the idea that artificial intelligence is simulation of cognition, is not autopoietic, it's a sort of allopoietic approach, according to the definition of autopoiesis and allopoiesis. Why? Because these systems are built by programmers with explicit reference to cognitive parts. So we have parts that are organized, but they are not parts that are self-maintaining. or self-organized, or at least not fully in this direction. The neural network approach, or the connectionist approach, that is maybe most probably the biggest part of artificial intelligence today, apparently is much more promising within the view of autopoiesis, because we have network organized with topological features. Remember the definition of autopoietic machines, especially in case as the recurrent neural network and also other similar kinds of deep network or neural network. There is a cognitive performance, even though the neural networks are used for a very big range of performance. But cognitive performance could be found in the categorization, recognition, pattern recognition, sensory motor action that are very well performed by neural networks today. Among different other kind of tasks. And there is the network organization that is very relevant for the performance. So this is autopoiesis. Is this autopoiesis? No, it is not autopoiesis to me, because the cognitive performance is emergent behavior after training, and we don't have the notion of self-maintaining or self-producing. There is an approximation to the best performance after the different phases of training of a network, but it's not the construction of a new part of the neural network in the main field of neural network. So how can we use other approaches, different approaches, approaches that I call chemical AI, stay in the main line of this this workshop. If we consider that the most important cognitive approaches to AI that we have today are at the same time organizational but not non-autopoietic, we can find that we can think that these facts seem to divide living systems from cognitive systems from the point of view of autopoiesis. So autopoiesis is related to living systems and non-autopoiesis is related to cognitive systems. How can we put together these two parts? Should we consider maybe chemical features of AI systems to bridge the gap? What is what I call chemical or biochemical AI? But we can find in the past some very well-known approaches to artificial intelligence that are close to chemical approaches. The study of Turing on morphogenesis, the study of von Neumann on cellular automata that we can trace back to the 50s of the last century. The other approaches on complex system, evolutionary algorithm, evolutionary programming. These are all attempts to logically define and computationally exploit the laws of evolution at the very base of life formation. But these are history-dependent phenomena. So it's not in the field of the autopoiesis. It's not organizational or organization in the sense of autopoiesis, for at least in a full way. A last embodied chance that I want to suggest is the swarm approaches to artificial intelligence, the swarm intelligence, cognition, robotics systems are a class of bio-inspired artificial systems that are inspired also, not only, but also to one biological structure, that is the superorganism, especially, for example, the ant colony. This is the maybe well-known book by Holdover and Wilson on the superorganism that was published maybe 10 years ago or something like this. Why this is a last and body shine? Because an ant colony is an autopoietic organization. It's a living system that we can analyze from the point of view of the life and the point of view of cognition. Why? In this in an ant colony, we can find unity, network, chemical relation, because the stigmergy is the mechanism by which agent environment relationship take place, and they are based on chemical relationship, and life and cognition. It has the parts and their goal-oriented, cognitively understandable functions of the superorganism are strongly related, but with a very big caveat, life and cognition are referred to the superorganism. Not, they are not the life and cognition of the organism that are its parts, so the ends. Now, this is very interesting to me, because maybe we can find a connection within the view of the autopoietic organization, if we use a weakened, maybe deflationist notion of biological unit to better characterize the relationship between life and cognition. So we can see that an ant colony has a life with self-maintaining, reproduction, and the research of a nest, and that is, we can analogy to skeleton in maybe human being, but we can also see the action of the ant colony. We can observe them as cognitively oriented. So the search for food, the search for a nest, the search for maintenance. We can describe, even though the description is not a good term for in the autopoietic view, we can see, we can understand the behavior of an ant colony also from the point of view of cognition. So my conclusion, because I'm almost out of time, I wanted today to analyze the notion of autopoiesis and its relation to cognition, that is in the, for example, in the book of the 1972 are connected to the nervous cell, so to the brain and to the nervous system. So this is the why, how they connected Maturana and Barrera, the autopoiesis to cognition. Autopoiesis, and I wanted to stress also that autopoiesis is strongly based on organization. There are, and then I mentioned the fact that the current approaches to cognitive AI are, at least in some relevant cases, organizational, but not yet not fully autopoietic. If we consider some of bio-inspired artificial intelligence approaches, some that I call chemical, because they are they could be considered close to the chemistry or chemical relationship and organization, and their model and situation, I think that they are good candidates to connect life and cognition within the autopoietic view. So the next and the example I gave is one of the superorganisms. So the next in their simulation in the swarm approaches. So the next step, I think, could be in this this line could be exploring the theoretical, but maybe maybe also not theoretical, to me theoretical consequences for synthetic biology, because it was the other part, the other flip of the coin that I could not analyze, because I pointed out just these aspects in connection, in which I want to show the connection between autopoiesis and some current approaches to artificial intelligence. So, end.
Okay, thank you very much, Francesco. And now there is time for questions. If someone would like to ask question, just raise the hand or write in the chat. I have one question. It's very intriguing to me what you said about neural networks, which have, let's say, some organizational properties that would make them a candidate for cognitive system. But you said that at the end they are not autopoietic. And since I am actually working on the design of chemical neural networks, so my question is, what would be, for example, the property of a chemical neural network that need to satisfy in order to match your criteria? For example, if you imagine that the nodes of the neural network are molecules, is it sufficient that the system produce new nodes, for example, produce the same nodes of which it is done?
Okay, thank you. This is a very good question. I think that what lacks in the neural network view to be a very autopoietic system is not a part of cognition, but is the part of life. They are not built to as the life systems in the autopoietic view, but I think that maybe they could be. It's not one goal of the neural network approach, but maybe it is more. It is easier to arrive to a fully autopoietic view through neural network or deep neural network, chemical or spiking neural network, than with the classic approaches, the other one, the cognitive approaches. Still, I think that we are not now in that situation. And also there is another problem. Neural networks are not totally explainable in their performance. So we have an emergent performance, but we cannot explain all the parts usually of the network that are performing. Why are they performing that particular task? So,
okay, so since we are speaking about neural network, there is Juan Carlos comment. Please, Juan Carlos, you can activate. Yes.
Yeah. No, I want to introduce that in the theory of autopoiesis, that has two branches. The other is biology of cognition, who is the origin of the theory and is about the nervous system. And the important factor is what is called operational closure, in the sense that the network acts, confront a universe, changes his configuration, and acts again. So is the loop of action perception loops that is the fundamental aspect of having a self-referential network. And this is the most important thing in the theory. And I will explain these aspects in my two talks. But you can get a network that is self-referential in the sense, has operational closure, which is the most fundamental property according to the theory. Not that the fact that they are made by neurons that are cells or not. The important thing is this operational closure that defines the trajectory of the network.
Okay, thank you very much for these remarks. And so I think that is another.
Luisa, maybe?
Yes, I totally agree with Juan Carlos, and I wanted to make a question about the notion of organization you use is because from the point of view of the theory of autopoiesis, the biological organization is also not only as this main feature, the closure, but as this main feature, the closure, because it's in a relationship of complementarity with the structure. So it's true that the organization is the invariant of the living dynamics, but because it's continually produced through the construction and the deconstruction of the components, so through the structure, through the concrete aggregates of components and the concrete operation of construction of the components. So it's a concept of organization that doesn't leave space to functionalism. There is the multiple realizability because you may have different structures that can realize this kind of organization continuously, but there is no space for an independence of the organization from the material realization, because there cannot be such an organization without a material organization, a material structure continuously producing it.
Yes.
So I was wondering in what sense the form of AI you were describing, according to you, are organizational, because this is, in my perspective, is clearly not the autopoietic organization they are displaying. Yeah. It's another form. So Maturana can talk about the organization of the chair to introduce the idea of autopoietic organization, introduce the idea of organization, but clearly there is a difference with the autopoietic organization. So I wanted to understand what you mean when you define these forms of AI as organizational. This I really didn't get.
Yeah. I think that these two kind of system, the cognitive architecture and the neural network approach, are a system in which there are parts that are connected in an integrated network, and they are also a unity. So this is close to the notion of organization of autopoiesis, even though they are not.
So only this aspect then. So is the interconnection and then the network like.
Yes, and it lacks the part of agent environment relationship, but we can also analyze, especially neural network, from a dynamical point of view. So there are many points of contacts and differences, and I think that if we can eliminate the differences or the point in which these systems are far, we can go beyond some obstacle, some limitation that today we have in this system. So this is my suggestion.
I think it's very interesting because from the neuron networks of the networks of ideal neuron from McCulloch and Pitts to the neural networks of today, there is this idea of network of organization as a network that is coming. But then the autopoietic network is something that comes that generates a new dimension in this. But it's still as rooted in McCulloch and Pitts, because also Maturana worked in the same group and stuff. So it's very interesting. So thank you very much.
Thank you.
Okay, perfect. So thank you again, Francesco. Let's move to the next speaker. So we have a little bit delay, but actually we have time because we really have enough time till 1 o'clock. Okay, so next speaker is Takashi Kegami. He's very well known to the artificial life community. So I invite Takashi, here, perfect, to share the screen, and please, you can start your presentation, half an hour.
Okay, yeah, thank you. Yes. Can you see my slide?
Yes.
Okay, can I start now? Okay, thank you very much for inviting me to this workshop. Actually, I really want to come to Trento, but yeah, maybe next time, next year. Anyway, I want to visit Trento to see Martin and Richard. Probably he's there. So this my talk today is about it's not about the synthetic biology, but it's more about the real biology. This is the title is the collective memory of tetrahymena populations. I'm working with Hiroki Kojima. He's my postdoc, and also Akiko Kashiwagi, and she's a biologist. This is the experiment that I'm going to introduce today. And Itsuki Doi, he's also my postdoc, working on the analytical side of this work. Okay. So first of all, so this is a synthetic biology. I did with it's a very simple experiment with it is state with the solution that you can see this interesting collective behaviors of oil droplet. And then interestingly, some of this flat droplet moves abruptly, from right to left. And then, but some of the circular ones is moving. Some of them are moving periodical way. Some of them are just stay there or moving with a constant speed. But you see some pink flat one is chaotically oscillating. And I really like this. I've been studying this one with Martin Hanzik for about 10 years. Then, as Stuart said, whether maybe this is life, but I think this is not the life, right? So I'm not so much satisfied with this not life of life behavior, right? So I try to look into the real biological collective motion to see what is missing here, what is lacking here, and then maybe by understanding the missing piece, maybe we can generate something more adequate to the collective intelligence. That's what I'm going to talk. So my talk is about this tetrahymena collective motion. So it's about, so this scale is about 800 micrometers, and each one is about 40 micrometers. So it's moving around, and some interesting collective behaviors. I don't know whether there is a swarm or not, but that's something I'm going to talk about today. And the experimental setup is very simple. So we use this microscope, and then heating up the glass plate at 35 centigrades. Then we culture the tetrahymena thermophile of the strain SB210. And as you see, there is a silicone oil and then PDMS microchamber that we found. We set up, then just observe how this tetrahymena moves around, right? And this is the image of a stereo microscope. Sorry. So then microscope, under the microscope, that you can see these tetrahymena moving around in this way. Then we can use either deep neural network or some algorithm that we can identify each cell, and then can identify the individual and tracking all the individuals, how they move around as time goes by. Then we can identify what the behavior, how the behavior changes over time, and then how they are interacting with each other. And if it divides, and then when it divides, and then how the behavior changes over time. So this is possible in these days by sort of machine learning and other techniques. Okay. And one of the big questions that I want to ask here is that even with the same genotype or the same genome of the tetrahymena, behave quite differently at the individual level and then also macro level, because we put tetrahymena in the well and then moving around in the well. But the behavior is very different from one to the other. For example, like this one and this one. So on the right-hand side, the individuals are moving more rapidly and more, energetically, I think. But this one, sorry, the movie is sometimes stopped. But on the left-hand side, they are more quiet, and then they don't move so much, energetically. So what makes difference is the big question. Even with the same gene and the same numbers of the individuals in the same well, and I think the environmental condition is almost the same. However, the collective behavior is different. So maybe the interaction matters, or some other effect is there. So we know that tetrahymena can sense the chemicals in the environment, also the chemicals secreted by the other agents that the tetrahymena can sense. And by sensing the chemicals in the solution, either the individual are attracted or repulsive from the other agents. That's my understanding. Then, so that we expect that there is an interaction between cells, right? So first of all, my big hunch from the beginning is that what is the candidate for a phenotype of tetrahymena? So I thought, okay, maybe the distribution function of the kinetic energy is that can be the candidate of the phenotype of tetrahymena. So this one is a single agent in the well, so that he's moving around, and then we calculated kinetic energy of those individuals. And the vertical line is a logarithmic scale of a histogram of the kinetic energy. And then the horizontal line is the kinetic energy. Then you can see the straight line implies that the behavior, the distribution function is exponential curve. Right? And then after 1 hour, still the behavior is exponential, right? So what does it tell us? This exponential curve is a good phenotype. One of the, I think intuitively you can understand that if exponential curve emerges, that means the distribution function of kinetic energy obeys the Boltzmann distribution, means that ensemble, this is like a Brownian motion. So the tetrahymena is a Brownian motion, which I thought was not so much interesting, because it's a Brownian motion means it's random behavior, and it's randomly colliding each other. So that even if there's an interaction, but the interaction doesn't so much affecting the behavior, right? However, the distribution of kinetic energy does not simply obey the Boltzmann distribution all the time. Sometimes, as you see, these are the different samples. Some of the individuals shows some heel form potential, and then sometimes they have some different curves. So maybe this kinetic energy distribution can be the potential for the phenotypic pattern for the tetrahymena. And then I want to call it individuality of tetrahymena, taking this exponential. It's not a non-exponential curve of those kinetic energy. Then we increase the number of tetrahymena in each pot, each well. So when we increasing the numbers of the tetrahymena in one single well, So on the left-hand side, you have 4 cells, and on the right-hand side, you have 8 cells. Well, for the 4 cells, well, we still have like a straight line like behaviors. So maybe these ones are more like very much close to Brownian motion, I think. However, on the left-hand side, on the right-hand side, you can see there's some convection, right? Becomes convex downwards, and I don't know whether that means, but certainly there's some effect when you have more than 2 individuals in the same system. So we computed the Jason-Shannon distribution of 2 energy distributions. So assume that these are the P and Q, and once you compute Jason-Shannon divergence, then which means that the P comparing with M is P plus Q divided by 2, and then and extending P and Q, then this formula that you can calculate how much similar, the degree of similarity of 2 distribution functions, P and Q, right? And DKA is a Kalbach-Leiber divergence. So by using this, that you can see how much is the difference between 2 distribution functions. So when I do this, this is a pseudo distribution function. You can just look at this on the right-hand side, that the random behavior, randomly picking up the single agent, it's not actually interacting with others, but superposing on the movie. Then this when the time goes by, then Jason-Shannon entropy goes down, divergence goes down, means that behavior gets similar when the time goes by, right? But when there's a number of cells is 2, 3, 4, 5, 6, 7, 8 in the same cell, then the distribution becomes rather diverging. Means that the shape of the distribution of each individual becomes more different over time. In the absence of interaction, which is on each left curve, on this pseudo pattern, then the opposite converges. So if there's no interactions, then the Jason-Shannon divergence is about 0.1, and then also decreasing over time, right? But when tetrahymena is interacting with each other, I think, and then if it's not random, then the distribution function is either diverging. That's one of the discovery. And then this is a swarm plot of the paired Jason-Shannon distributions. For when there's a number of cells is 2, 3, 4, 5, 6, 7, 8 cells in the same cell, same wheel, and then individual behavior is rather diverging. But as a well, the individual of the well is rather decreasing. So what this one tells us is that the shape of the distribution per individual evolves into different distribution as the number of cells in the well is increased. However, the average divergence is rather decreasing. So as a well, the individuality is losing, but as for the individuals, because of the interaction, the divergence is rather the behavior becomes different. That's something that we found from this experiment. Then we switch on to the case where the cell can replicate. So when the cell is interacting with each other, and when we identify and tracking the cell, then at certain time that they can replicate. see the cells movement is slows down, and then they start to replicate. Again, it goes, now this green one is going to replicate. So that's what the process of when it's not a fixed number of cells from the beginning, but when the cell is dividing as time goes by, and that's what we see in the system. So we have a phylogeny of time series from the single cell. From the left top divided, so this is a 2 time series that emerges. Then after like 100,000 time steps, then again there's a 4 time series. Again, then it goes to 8 time series. So division of cellular systems is now observable by looking at the time series. And behavioral heredity, what I call heredity over behavioral heredity, is now measured by the kinetic energy distribution as we did with the fixed number of agents. So for the on the left top figure, that first of all, this is like this one. Then it goes to 2 different curves and 4 different curves and 8 different curves corresponding to the offspring and offspring of the offspring and offspring of the offspring of the offspring. So the 2nd generation and 3rd generation, 4th generations that we can calculate energy distributions because we thought maybe one of the first candidate of the phenotype is the kinetic energy distribution, right? And this is another example, like this one goes to 2 distributions, 4 distributions, and 8 distributions. And you can see the shape is similar, right? Even though the genotype is the same, but the phenotype is different. So the phenotypic different variation is inherited to the offspring, right? That you can see from this figure and then also from this figure. And then also when you see this right-hand side and left-hand side, the energy distribution curve is totally different, right? One is concave in downwards, and the other one is rather, there's no concave to the downwards, but it's something different, right? But as when the cell divides, this energy function curve is inherited to the offspring. So that's why I think there's a heredity in terms of phenotypic patterns. And this is the similarity of kinetic energy in a phylogeny. By taking the first one is the 1st generation, and next 2 column is 2nd generation. Next 4 column is the 3rd generation. Next 8 column is the 4th generation, right? When you are comparing with 2 to 3, and then this color panel corresponding to the Jason Shannon divergence. So how much their energy distribution is similar, right? And then when it's black, it's very much similar. And then when it's bright, it means that their distribution is very different. So when you see from here on the left top corner, you can see there's distribution there are heredity from the 2nd generation to 3rd generation to 4th generation, right? But for example, like this one, from the 2nd column on the right, there is no heredity, that there's no trivial heredity exists, right? So some of the cells is very good at transferring their phenotype in terms of energy distribution to the offspring, but some of the cell cannot transfer their phenotypic distributions. So there is something that exists that determines that this heredity possible or not. Then my final question is, what is the difference between the population created from the common ancestor, and then finally becomes 8 cells? It's a 3rd generation. And the population created by the 8 cells, just extracted from the clone soup and put it there, right? So this one, I don't know whether it moves. So as you see on the left hand side, it's created from the same the common ancestors. On the right hand side, this is just random, putting extracting from the same clone soup and make 8 cells ensemble. But you can see, I don't know whether you can see the differences, but once you observe it longer and longer, that you see something is different. So we try to characterize the differences. And when you have the unrelated cells in the well, as we saw previously, that the JSD Jensen-Shannon divergence it's around 0.1 for 7 cells and 8 cell cases. But for this cell from the common ancestor, this is way below 0.1. Means that when the cell is from the same common ancestor, then their energy curve is sort of similar to each other. Means that there is some heredity exists. Therefore, their offspring and offsprings are kind of similar phenotype in a sense of kinetic energy distribution. But when there's unrelated cells in the well, even though there's an interaction, but still the difference is big comparing with the cells from the common ancestor. Then when you see one of the examples from the common ancestor, these two tetrahymena shows very interestingly synchronized behaviors. Even though I don't know what kind of interaction that makes this kind of behavior, but only when you see example from the common ancestor, you can see those individuals shows the very similar behaviors rotating with it at the same time. Not very much close, but a little bit closer to each other, they're making rotating behaviors. So we try to analyze this kind of behavior by computing Jensen-Shannon divergence, but this time not only speed or the kinetic energy, but also a rotating speed, right? So we have to calculate angular velocity and also translational velocity, 2 of them. Then the distance between the time series of this one and this one is measured by the 2 points, right? V and W. Then we projecting onto the multidimensional scaling, the 2D space, and see what we can see this. So when there's a collective, and then there's individuals. Let me see. This one is much better, right? So when you see the blue one is from a single cell, common ancestor, and the yellow one is just put them from unrelated cells from the same genome soup, right? So apparently their behavior is so different. Yellow one is unrelated cells, more converging around the center, but cells from the common ancestor is more diverging in the space. Means that something is different between cells from the common ancestor and then cells, even though they share the same genome, but they are not from the same cell. So my discussion here is that I think we have 4 different discoveries. One is there is a variation from well to well. So each well shows different behaviors. So the variation of individuals within the well is even greater, right? So the individuals within a cell, that's some kind of interactions, right? But then there's also variation from well to well. Every individual has the same genome, but because of the plasticity of the phenotype, and this phenotype variation is also different from one to the other. That's something is quite interesting, we think. And then the population of 8 individuals derived from the common ancestor, and the population of 8 individuals randomly sampled from the clone population appear to be different. And then I think because some sort of heredity is conserved when there is a population of 8 individuals from the same common ancestor, but there is no such things exist in the randomly selected egg cells. That's why. So we expect that the memory is maintained in the interaction between cells, but not always. That's the one of the things we think is interesting. And the individuality of the cell, first cell, determines the individuality of the well, because when they dividing and making a second generation, third generation, fourth generation, and fifth generation, all the individuals are from the common ancestor. Because they are coming from the common ancestor, they are sharing the same property. So that once you see this individual as a whole has some sort of individuality, which is different from well to well. But once you make a cell, the well of the assembly, not from the common ancestor, but just randomly picking up from the genome soup, then their behavior is from well to well is relatively similar. So that's what we think is interesting. So the individuality of the cell is still diverging, but once you make a cell, once you're looking at the macro level, then whether the assembly is from the common ancestor or not determines the individuality of the entire assembly. So also this phenotypic variation is there's it's a previous work by Rivera and Livera in 2014, but they tried not. So what they observed is something that the individuals should not interact with each other. So it's more like whether the individual cell has some similar property of its parent or not. That's what they studied. But from this study, we think even in a strong interaction regime that we study here, is still, and then also rather enhancing the heredity when the cell is coming from the common ancestor. But this interaction between cells is not synchronizing with each other. As you can see, from the cell is randomly picking up from the common soup, their behavior becomes rather different. It's diverging over time, means that their interaction between cells is not synchronizing, but rather they're synchronizing with each other, I think. So the interaction between the cell is much more complicated than we expect. The interaction can be attracting or repulsing or synchronizing or desynchronizing is depending on how they are sharing phenotypic variation, phenotypic type or not is I think one of the hints that we can find here, but still we need more to study to answer those questions. Okay, thank you very much. That's my talk here.
Thank you. Thank you very much, Takashi, also for being perfect time. And this is really interesting results that reminds me a little bit the results from Eshel Ben Jacob from Israel, who took bacteria growing on hard surfaces and showed that they can, let's say, transmit some phenotypic behavior when separated from the colony. Okay, so there is time for questions. Any questions here? Raise your hand or write it in the chat. Okay. Takashi, I have a question. Maybe there is Seth.
Yeah, I have a quick question.
Please.
I may not have understood completely, but so the numbers of cells in your Petri dishes is still relatively small. Do you think that a cell that has derived from a long sequence of reproductions, and so therefore may have changed from its ancestral cell, if it was placed back in a environment with a lot of the clonal ancestral cells, do you think that would revert its behavior, or that it wouldn't be able to express the behavioral variety that you have seen in the results that you have shown?
Yeah, that's a very interesting question. But because we looked at second, third, and fourth generations, right? And then there's some heredity is at least some of the cell can sustain at least until fourth generation, right? But once we see more longer and longer chains, maybe their heredity cannot be maintained. But as you say, once you bring it back to some ancestor cells, or coupling with some parent cells, and then maybe we have to decrease the number of the cells. If we can recover the heredity, that might be very interesting. But it implies many interesting things, right? Whether we can like retrieving the memory of the ancestor phenotype, or this kind of things is possible. So that's why I think it's a very interesting idea, but we haven't tested it yet. So yeah.
Sure. Thank you.
Be nice. Yeah. Thank you.
Other questions? Takashi, do you think that when a cell divide and makes two other cells, there is something in common beside the internal genome? The common thing is that their position and their, let's say, velocity. And therefore, they experience very similar environment in the very early steps of after division. So do you think this somehow, play a role in the observed behavior?
From the dynamical systems point of view, what you're saying is totally right. So what Pasquale says is maybe right. I mean, because they see the same view right around it, and then also maybe their initial state is very similar. So yeah, you're right. So even though when they separating with each other, their memory is slowly decaying, and then also the minute differences is amplified when they're moving around. But that's why. But because of the synchronization, behavior emerges, because as you said, those cells share the same initial configuration when they divide each other. Yeah, you're right.
Thank you. Oh, Yutetsu, please go ahead.
Thank you very much for a very interesting talk and very interesting experiment. I don't know if my question makes sense or not, but I want to ask, your experiment, when you start from the single cell and this single cell divided, finally goes to the 8 cells. So the first one is only one cell, so that he cannot interact to others. However, after the self-reproduction, 8 colonies can interact with another cells. So is there any difference in the moving pattern? Because all of the cell has exactly the same genome, and the place environment is exactly the same. Only difference is there's a partner or alone. So how they change the moving pattern?
Actually, that's what this figure shows, right? So this is the first generation and second generation, and once if it's totally black, means that their behavior is completely the same, right?
Okay.
The first generation is different from the third one and fourth one in this figure, right? But,
so what's happening in the third generation? So the third generation and the fourth generation is quite different.
Right. In this case, yes.
Yeah. What's happened?
I don't know what happened. maybe some in terms of dynamical systems. I don't know. I don't think this one is a complete dynamical systems, but maybe attractor is shifting from one. If they have 2 attractors, shifting from one attractor to the other attractor is one idea. Or maybe they are creating new attractors by interacting with each other, because this degrees of freedom is going up, right? So by interacting with each other, they can make a new internal state. That's why they can, dividing into different ways.
So you mean the attractor is also growing up?
Yeah, attractor is growing and then differentiating. So first of all, maybe they have only one attractor, but once they're dividing and interacting, they can create new attractors, or maybe something like that may happen. But so we are looking at RNA-seq single-cell RNA sequences, try to understand whether how many internal state they have in terms of RNA, sequence. So that's something I'm doing now. So it's a very interesting question. Thank you for asking me this question. Yes. So how many numbers of degrees of freedom they're increasing is something interesting. Yes.
Very, very good. Thank you, Takashi. Okay, and now we can move to the next speaker, who actually is the protagonist of Artificial Life 2022, which is Sylvia Holler. Okay, Sylvia. So you can start your presentation. The authors are Sylvia Holler and Martin Ancic, but Sylvia will present. Please go ahead.
Yeah, thank you, Pasquale, for your introduction. So, 1 second. Okay, so what I will speak about today is artificial cells as biological cell transporters. So as already showed you before by Takashi, we even worked on droplets. So we work on droplets, and these droplets, as shown even by him and even other people during this week, such for example, the keynotes that we had, Yob and Dorotang and these kind of things, sometimes show some lifelike behavior. So they aggregate, they show the composition of compartments that seem similar to cells, or show some behaviors that are lifelike. In this presentation, I will focus on one life behavior, that is chemotaxis. So as probably many of you know, Martin started this work a lot of time ago and showed that droplets can show chemotaxis. So as you can see from this video, we can see, for example, dictyostelium that is following a gradient of CMP and is reaching its source. Similarly, what Martin showed is that yeah, I'm sorry. It's normally answering more fast. That even chemotaxis droplet, if a chemical gradient, that is the one that here is created in purple and is a salt gradient, are able to follow the chemical gradient and reach its source. So even in this case, we have a chemotaxis. So chemotaxis is shown by natural system, but even by artificial ones. The system that we are using and speaking about is a droplet system that is composed by a body that is mainly composed of decanol, and one environment that is water with high pH between 11 and 13, and one surfactant that is decanoate. There is no specific onboard fuel in our system, but there is one energy gradient that is created by the environment in which we are adding a sodium chloride solution. So we are created a chemical gradient that is created one energy gradient, and for energy minimization and surface tension minimization of the system, the droplet is moving towards the salt source. Yeah, so as said before, the droplet moves to minimize its energy. And here, for example, some plots that shows the surface tension minimization upon sodium chloride addition inside the environment, so the Petri dish in which the droplet is moving. So for this kind of system, as you probably even know for this part, Itkachikova, for example, in 2014 already showed that she can create 2D mazes in which the decanol droplet that you can see here in pink move towards other type of droplets that in this case are reported in yellow and that are releasing some salt. And so even in this case, the droplet in pink, so the decanol one, is following a salt gradient and is somehow able to reach its source in a smart way. So in one artificial intelligence-like way, without the presence of almost anything. So there are no cells, there is no brain, but however, these kind of droplets are, due to this energy minimization, are able to somehow find inside this maze the salt source. So starting from this previous work, we choose to integrate this system and to try to use it as a transporter. So we tried at the beginning to transport inorganic objects as shown by this video, and we tried to transport, for example, here a piece of copper wire. So the solution that you are seeing to be added to the system that is in blue is a solution with high content of surfactant that is somehow destabilizing the coupling between the decanol droplet and the copper wire. So as you saw from the video, we were able in this case, for example, to move the droplet paired to the copper wire to then during the time to somehow decouple them one from the other, so decouple the droplet from the copper wire, and then when we specifically wanted it after 2 movements to induce the release of the copper wire from the droplet. At this point, so we transported inorganic objects, so we were thinking about what about to transport even organic objects or cells. So when we deliver a package, we would like to arrive sane and safe to its recipient. And as said before, the system that we are using, it's a high pH system. And inside this kind of high pH system for cells, for bacteria, for mammalian cells, it's even for yeast, it's really difficult to survive. So we try to encapsulate these cells inside some specific package with which they could arrive sane and safe to their final destination of the chemotaxis. So what we did is to use alginate gel and cross-link it to create some capsules with which the capsules could have been moved. So in this video, where is pointed by the arrows, there are 2 capsules that are placed upon the decanol droplets, then and in which were previously incubated some gram-positive cells. So here instead from the circle in light blue, you will see the place in which we want the capsules to arrive through chemotaxis. In fact, we will now create through the addition of sodium chloride solution 1 chemical gradient. Now the couple of decanol droplet and the capsules are moving. They are decoupled as before from the capsule using some high content surfactant solution. And then we can easily move the droplets for a second times or how many times we want to other destination, this leaving the cells inside the capsules free. So upon this transport, the cells are surviving. We are able to transport using this kind of system both to gram-positive, gram-negative cells and even yeast. So this was a really nice result, but we wanted to try to transport even other different type of cells. So we tried to transport mammalian cells, but as you can predict, it was at the beginning really difficult because the conditions of the transport are really difficult for mammalian cells. Mammalian cells normally live in specific pre-prepared media in which the pH is not that much varied from 7. So if before, for example, from like bacteria, we were picking up the capsules and the cells were easily surviving, this did not happen for the mammalian cells. However, while trying to transport them, we discover that we were able to transport them in another medium. So we have a new dynamic droplet system in which we have the environment, the body that is a new time, the decanol. The attractant that is always the sodium chloride, the cargo of our capsule that is alginate capsules that So if in the systems before it was transporting bacteria, now instead it's transporting mammalian cells, different type of mammalian cells. And now what is varied, as you can see from even from the color of the solution incubated inside the Petri dish, is the environment. So what we were able to transport the mammalian cells with were DMEM, so the pre-made media in which mammalian cells grow, in which capsules were incubated with cells for 2 days. So this is, for example, the video of what we were able to do. So as you can see, the system is similar to before. We still have the decanol droplets, we still have the capsules, and we are able to transport them. So what happened during our experiment is that we found out that if we're leaving the cells incubated inside the alginate capsules, inside the DMEM, that is the media in which the cells normally grow for 2 days, they were somehow releasing something that probably was a molecule, an amphiphilic molecule that was acting like a surfactant that was enabling the chemotaxis inside this new media, this pink media. So this is how the general transport experiment worked. So we had living cells, we were encapsulating them inside the alginate gel as shown before already for the bacterial cells. Then, however, somehow during the incubation, the mammalian cells are releasing some surfactant-like molecules, some amphiphilic-like molecules. Here they are minimized to simply charged head group and unpolar chain. But this release of this surfactant enabled the capsules to remain attached to the decanol, and at the same time to have one organization of the surfactant surrounding the decanol droplet that was still enabling the chemotaxis, and so the surface tension minimization and air energy variation to which the chemotaxis itself is due. So what we tested at this point was the association. So when we started to try to transport bacteria using this system, it was really difficult at the beginning to couple the hydrogel of alginate with the decanol, because you can imagine it's really easy to understand it, because however the decanol it's really hydrophobic, and instead the decanol, the alginate capsule is mainly composed by water. So to enable the coupling between the capsule and the droplet, we had, for example, to integrate inside the alginate gel some surfactants. What happened here in the case of the mammalian cell is that, however, during the encapsulation of 2 days in the mem of the capsules, as I said before, the capsules are somehow, the cells in the capsules are somehow releasing some surfactant, and these retention of the surfactant is enabling the coupling between the capsule and the droplet. So as you can see in the plot, for example, we have almost no retention of, for example, capsules of alginate suspended in water or DMEM, or in the water phase in which bleach-treated cells are pre-incubated. But instead, in the case in which we have the resuspension of the alginate inside supernatant dilutions in which cells probably releasing surfactants are incubated, we have a super higher retention time of the capsules on the droplet. So we somehow find out that coupling the capsules with the droplet and coupling the cells with the capsule, we are increasing the functionality of the capsules itself, because if before we had to integrate the surfactant inside the capsules to transport, for example, the bacteria, now the mammalian cells were on their own releasing the surfactants. So here is, for example, a survival plot. And so we check the viability of different type of cells before and after the transport. So the cell lines that better survive this transport were A549 cells. And A549 cells are, for example, on cell line derivated from lung cancer. So these kind of cells are even thought to probably release mucins and other kind of surfactants that are normally found in the mucus inside the lungs. So we could somehow predict this release of surfactant inside the water phase in which they are maintained. However, we were able to somehow transport other type of cells that were somehow surviving, but with less viability, as for example, H460 and H1299. So, however, what we tried to analyze better at this point was the variation of the surface tension due to the surfactant release inside the water phase that was due to the incubation of the cells inside the capsules of these lung cancer cells in the specific. So what enables the chemotaxis is the variation of the surface tension. And in this plot, you can see that only the water phase of in which capsules that are alive and there are these lung cancer cells are enabling this variation in surface tension that is giving the chemotaxis. So for example, in the first line of this plot, you can see the normal chemotaxis that we shown for bacteria that is working, and the variation, for example, for the decanoate with pH 11, decanoate with pH 7, and then the variation for A549 capsules in demand for 2 days, H1299 capsules in demand for 2 days, and H460 in demand for 2 days. Instead, if we simply try to check for the surface tension upon salt addition in demand only, we almost have no variation. The same is for the water phase of bleach-treated cells incubated in capsules. So then we checked even for the velocity and the efficiency of the transport that was correlated to this surface tension variation. As you can see from these two plots, we have, let's say, two different populations. One on the bottom left of our plot, and the other one of the top right. The one of the top right is one that has high surface tension and high velocity, or high surface tension and high efficiency, and is the only one in which the chemotaxis is effectively working. And in fact, all the points that we can find in the right top spot of the image are decanoate pH 11, decanoate pH 7, decanoate pH 11 with capsules even without cells. So in all these cases, we have one surfactant that is given from the outside, from our hands, adding them to the water phase. And then in the last case, for the blue spot, we have lung cancer cells with supernatant and capsule water phase. So even in this case, we demonstrated that this lung cancer cell water phase has somehow something that is not simply enabling the chemotaxis, but it's even increasing its velocity and efficiency. So as shown before, not all the cells survive the transport. So to enable the transport of more cell lines, and even cell lines less easy to cultivate than A549, we tried to double encapsulate our alginate inside our alginate capsules, our cells. So for example, we double encapsulated EC293 T cells and even some fibroblasts, HS68. So what we obtained is that using a double encapsulation, we were able to transport even this kind of less robust cell lines, and that they were surviving the transport. So to conclusion, what we were able to do was to integrate artificial system showing intelligence, because even as showed by Itka, if placed inside a 2D maze, one decanol droplet is able to find the salt source from which the salt is released, even without initially knowing where it is, with biological system, because we were able to transport both to bacteria, yeast, and even mammalian cells at the end. We saw that some cells augment their function while incubating inside the capsules, because instead, normally, if we, for example, take the water phase in which cells are incubated, in which A549 cells in the specific are incubated for 2 days and try to use it for chemotaxis, it doesn't work. It works only when the capsules are when the cells are incubated inside the capsules. So the capsule incubation is somehow augmenting the function of these lung cancer cells. At the same time, the surfactant that is produced from the cells can be used for the chemotaxis. And so not only the cells are increasing their function, producing the surfactant that is enabling the coupling between the capsules and the decanol, but they are even enabling and increasing the function of the water phase of the normal DMAM from the release of the surfactant itself. So we have in this way created a double-side symbiosis system that is at the same time boosting the functionalities of both two artificial systems, that is our droplets, and biological systems. And thank you for your attention.
Thank you. Thank you very much, Silvia, for this presentation about droplets, very fascinating word, and this interconnection between abiotic and biotic systems.
Yes.
Okay, so now there is time for questions. Is there any question? Maybe Chris Gordon is activating the microphone. Okay, in the meanwhile, I can make one question. Silvia, you told that at the beginning you tried with bacteria, but things were not so easy. And which were the problems? What happened?
Yeah, so the problem is that it was mainly between the coupling between the droplets and the capsule. So alginate gel is mainly composed of water. Inside decanol droplets is really oil, no? So they really don't like each other. So I had to do a lot of tests, and at the end, what enabled the coupling of the capsules with bacteria with the decanol droplet was the dissolution of the alginate. Instead, instead of normally doing it with water, we did it instead with decanoate. So we were adding to the water phase some surfactant. And so this surfactant was enabling the coupling with the decanol. Yeah.
Okay.
Thank you.
Yes, Takashi, please.
Oh yes. I have some stupid question, but
Yeah.
when you make a like cybernetic cell, because the cell you encapsulate into the like artificial membranes and things like that, right? Do you think you can increase the function of the cell, like speeding up the replication speed or amplifying the sensing the environment, or all this kind of stuff?
Yeah, so what we mainly tested and we find out was only the increase of the functionality for these lung cancer cells that were releasing the surfactants when inside the capsules, but if normally in their media, they were not doing it.
Oh, okay, I see.
So for these cells, it was found out. But yeah, one of the nice future perspectives is that, however, try to find that, because probably on other cell lines, even if we didn't find out any increase of function or better functionality for the chemotaxis, but probably for sure we could find other cell lines that could have one increase in other functionality. And I think this
that's very good. Yeah,
I think this is really interesting, because at the end it's all given by one interface interaction. So it's given all by the surface interaction between the cells and alginate gels. So I think that even in the future, it could give a lot of new insights, because at the end, varying the material on which you incubate a cell line, it could be probably vary somehow its function, no? So yeah, there is for sure a lot of new work and things to be discovered on this part.
Yeah, that's very interesting. Can I add one more question? So then my second question was, when I'm doing this tetrahymena interacting with each other, right? And it's the things that I want to know is real time examining which sensor is going to be activated and how they what sort of chemicals that they can secrete into the environment. So if you can, in a real time that you can record those things, that might be very interesting. But do you think you can like make a device that encapsulating this tetrahymena? Then when they're interacting with each other, that whether they can, real time, recording which channel is activated and what chemicals that they can absorb or they can secrete. What would you think? Is there any possibility of doing this?
I think it's feasible. I think there is a lot of work needed, but then however, like adding some fluorescent tags or some specific thing and with microscopy, I think it's feasible. Yeah.
That's very interesting. I see. Thank you.
You're welcome.
Thank you, Silvia. Okay, there is any other question? If not, we thank again Silvia. Very, very well done. And now it's time for our invited speaker, keynote speaker, and he is the Professor Juan Carlos Letelier from the University of Chile. Okay, he's connected. Okay, perfect. So Juan Carlos Letelier, University of Chile, Department of Biology, is a neurophysiologist, neurobiologist, and comes from the very long, important tradition of the Maturana and Varela lab in the University of Chile in Santiago. So Juan Carlos Letelier will present two topics. Actually, we have a long lecture of 45 minutes. And the first is about structural coupling and Bayesian inference, and the second part more on the historical side, the fundamental friendship between Heinz von Foerster and Maturana and Varela. Okay, so please, Juan Carlos, the stage is yours.
Perfect. Thank you very much for inviting me. And thank you very much for inviting me. And it's very early in the morning in the winter in Santiago, and it's cold and humid. Yeah, you must say in Italy you are hot and dry, or no?
Yeah, exactly the opposite. And we thank you very much for the, this very early time in Santiago. Sorry.
No, it's nice. Then the first talk will be a talk. You are going to see. These are talks. Myself, I don't do talks. I do performances. Then I apologize. Yeah. Let me find my. Then there. Do you see my screen?
Yes, perfectly.
Yeah. Then it's a question about the talk is something that you're going to see. I am doing it now at a high level of intensity, and it's an invitation for everyone. Yeah? Then I'm going to present what Maturana thought was his fundamental idea, that is the idea of a structural coupling. And I will join this idea with Bayesian inference. And I will explain the basics. And thank you for inviting me, and that's it. In this field, we have been surrounded by very brilliant people during the 20th century. We have Rosen, von Wexkull, who introduced the fundamental idea that all living systems are cognitive devices, Maturana and Varela, Kaufman with his autocatalytical networks, Eigen, we should not forget him with hypercycle, and Tibor Ganti, who was a Hungarian chemist who alone developed a theory that looks like the ideas of autopoiesis and the ideas of Rosen. But unfortunately, his work didn't make a big impact. Yeah? And then these are like the basic idea that today, if we want to do a real system biology that will produce a real living system, the thing that we were talked at the beginning of this session, we have like, these are the main elements. The main elements are the theory of autopoiesis, MR system, autocatalytic networks. Chemoton is a small population of models, but we always need to be aware that they exist, especially in the case of chemoton, because Ganti did a very precise analysis that we should always be aware. Hypercycles are by historical reason. I don't think that we will need them in the future. But without any doubt, what we will need in the future is the idea of Umwelt, that the idea that living systems create their own universe. That the objects that populate the universe are created by the living of living systems. And in general, unfortunately, biologists, we are like that. All these theories do not talk to each other. We never consider the other people, you know, we are very autarkic, we are self-contained. And the real reality is, my dear friend, the real reality is that our theories, you know, all the theories that I just presented are very interested and are quite seductive, but they're still very, very primitive. We are live in this universe of theoretical thinking about self-organization. We are like quantum mechanics in 1912, you know, before the atomic model of 1913, you know. We are just at the beginning. We cannot explain anything, very few things, you know. We cannot, of course, explain the origin of metabolic networks. My main problem is that we cannot explain the second point, the stability of the metabolic networks. You know, why metabolic networks are stable entities. It's a mystery. And also because a living system is a thermodynamical entity, our theories of self-contractions do not talk with thermodynamics. They do not talk well with statistical thermodynamics or with classical thermodynamics. That is very important. Yeah. Then there are many things that we cannot explain. Meanwhile, what I will call brute force approaches are quite successful. You know, molecular biology, that is really a brute force approach to understanding the universe, is able to sequence a complete human genome overnight. Imagine the amazing thing. You know? They can sequence 3,000 million base pairs of DNA in a single night. And they can edit DNA with this new amazing magical technology of CRISPR-Cas9 very, very precisely. And also they produce data in oceanic proportions. Now we know the metabolic chart of many different animals, the genomes of thousands. Yeah. And the other approach that I will call a brute force approach is what is called today artificial intelligence. That is nothing else, but is something that is looking for multivariate correlations in high dimensionality data sets. And with this, you do amazing things like to produce a machine that plays chess and Go at the level of a grandmaster. Yeah. Then these two technologies that are brute force, that are not really profound, they don't really illuminate the world, molecular biology and artificial intelligence, are really helpful for the scientists, what I would call, because I am one of them, the scientist that lives in a laboratory. Now they use these things. These two technologies, they serve to advance. And our ideas are seductive, but we don't produce real advance. Then I will explain you what we need to advance, according to then something very serious is missing in our theories, and I will explain that. And it's the relation between metabolic closure and cognition. First, this is my mathematics. This is cartoon mathematics. What you are seeing now is the cartoon of an autopoietic system that, because it's self-produced, it's the Ouroboros. It's like the, it's the Ouroboros idea. Yeah? According to the theory, and it's not, the original paper of autopoiesis distinguished two types of processes and components in an autopoietic system. The organization is the set of processes that do not change if the system must maintain his identity. And a structure is the set of components and processes that can change without changing the systemic nature or the systemic entity of the living system. Yeah, this is an important idea in the early theory of autopoietic system that usually is not, nobody underlines it. And I think that one reason is because the original texts were very obscure. Right? And then one consequence of this thing is that inside the living system, you have plastic and regulatory mechanisms that are an integral part of autopoiesis. This is extremely important. You have many components and processes that are only devoted to maintain the organization invariant, to maintain that part of the network, that part of the network, to maintain that part invariant. This part, you have a lot of hardware that the only purpose is to maintain this set invariant. And then it's, excuse me. Well, to give you an idea, in the bacteria E. coli codes for 4,401 genes, but only 2,000 of the genes, a little more than 2,000, they code for enzymes that direct metabolic reactions, to the real enzymes that direct the metabolism. The rest are protein that codes for signaling reactions, the reactions that are regulatory proteins. Then if you want to build an artificial cell, a very important point to have in mind is that regulation is not an afterthought, it is central. Yeah? If you want to build, regulation is central and it's very dense. Think always in Escherichia coli that has most of the proteins are devoted to regulation, not to metabolic control, not to be enzymes. Yeah, and here is, now I'm going to introduce what Maturana thought was his central idea. Maturana didn't think that autopoiesis was his central idea. Maturana thought that his central idea is the one that I depict here. I'm going to explain it, and it's called structural coupling. And the idea in A, you have an autopoietic system that is in relation with the medium, but the relation is just beginning to establish. This is why you have a cloud. This cloud is not that the autopoietic system has holes, is that the relation between the system and its medium is not well defined. But as the living system begins to live his life in the medium changes with the living system. Then this is why in B, you have now that the structure, this is what is called structural coupling, the structure of the living system begins to change as well as the structure of the medium. And then the unknown cloud diminishes. And in C, you have now a final situation where from the viewpoint of an external observer, you think that the living system has adapted to a very curious object in the universe. But the real reality is that the living system didn't adapt to the situation, to the object, but that the living system created the object in a mutual self-referential conversation with the medium. Yeah? And this phenomenon is called structural coupling. And this phenomenon, Maturana hinted at this phenomenon in the fundamental book or technical report we're going to see in the second talk of 1969. But he only clarified these ideas much later, around 1985. Yeah. And as I repeat, this was, according to him, his fundamental idea. Then if you take this into the context, then you have an autopoietic system that is now in a definite relation with the medium. You know, they have established this coupling, and then the living system. Here, I'm going to make a real important distinction. The living system is equal the autopoietic system plus its situation of structural coupling. You know, the living system is a relation with the environment. It's not the autopoietic system, but without the autopoietic system and its autonomy, you will not be able to construct this thing. It's fundamental. To be autopoietic is fundamental, but the living system is the relation co-created between the autopoietic system and the environment. Then, then a living system, in order to have this relation, he must have important subcomponents, you know, subsystems that are, every living system must have them. You are not going to be able to have a system if it doesn't have a sensory lamina, you know, a sensory surface where he can perceive its medium in some way, and another lamina, effector lamina, where he can act, and this acting can be acting upon itself, you know, not only in the medium. Then here is the important point. Every autopoietic system is inserted into an action perception loop. You know, and in this sense, every autopoietic system, even a small bacteria, and the wonderful experience that Takashi Sensei just gave, you know, with the movement, these things always are in an action perception loop, creating their structural coupling. And the big question is that a living system, the big philosophical question that every living system must answer in every moment is the problem of to act. Which is the action that I need to perform in the next second? The problem of living system is the problem of what to do next in order to maintain the situation of structural coupling with my medium. Then the fundamental, and here is the fundamental idea, and is that the sensory lamina, when the living system confront the universe, confront the medium, here is very important. Here, the living system confront the medium. In this confrontation, because of autonomy and because of the self-referential nature of the living system, the internal signals do not describe well the external objects. The internal signals, or the molecules that are created because of the interaction of the living system with the universe, with its medium, they are not, it's not a bijection. There is a very complex pattern. Then the living system must compute the following probability. Which is the probability that I am confronting the object X given that I have internal signal S, then it has to solve a Bayesian probability. Yeah. And with this probability computed, he will act, decide what to act, what to do. Then the fundamental problem of a autopoietic system with a structural coupling is to compute this Bayesian probability about the objects that he has built or is building because of a structural coupling. Yeah. A bacteria in every moment must surely be able to compute like 10 probabilities, but we compute millions. Yeah. This is why we have a nervous system. Then, in conclusion, at every instant, a living system, which is a relation between a autopoietic system and its homeworld, must compute a Bayesian probability, and here is our problem, which is a probability that I am confronting object X, given that the internal signals are S, in order to act. Thus, a useful theory that we don't have it yet, a useful theory of metabolic networks must use the following ideas. I think that is inescapable. We need to use the idea of metabolic closure in the form of autopoiesis and MR system and perhaps autocatalytic networks. But most centrally, we need to use the idea of a structural coupling with the ideas of Bayesian inference. We need to put elements of thermodynamics because living systems are, before anything else, we are thermodynamic entities. We are engines. We do use some fancy mathematics, like what I have published, this equation, what I call the Ouroboros equation, f of f equal f. You know, we need to use the fancy mathematics that Rosen dreamt. You know, Rosen was a man of intuitions, not a, yeah? Rosen dreamt about the use of category theory. We need to use modern category theory. You know, Rosen used a very old, the best, you know, the early category theory. Now category theory after 60 years or 70 years is a huge field. And there you have extremely powerful ideas to interpret many things. And also now I make the dark horse that was in my initial slide. I think that the ideas of Carl Friston about the FEP principle, the free energy principle, we need to begin to understand them. His papers are very difficult to understand, but he has, in a very convoluted way, he has this idea of a structural coupling and Bayesian inference. And an example of a very interesting effort is the paper given in this conference by Dr. Suzanne Steele called Inference, Prediction, and Thermodynamic Efficiency. There, this is the thing that we need to begin to think. And we need to solve this problem, we need a real interdisciplinary attack to this problem. You know, we need to do the things that biologists, they don't do. You know, biologists, every biologist, they develop a theory and they forget that other people is working in different aspects, in other theories around the same problems. No, we need to have right now an interdisciplinary attack to this problem. And I have some ideas that I don't want to do that. And I have even including funding. You will love that. Yeah. And this is a joke for my second talk that fortunately now, I am very proud of what Maturana and Varela did achieve, because now we can talk about autopoiesis in an artificial life conference. And we can talk a lot about autopoiesis in an artificial life conference now. But you must know that I went to the first Artificial Life Conference in Los Alamos in 1987. And I couldn't give my paper about autopoiesis. It was considered that it was too philosophical. Yeah. Then this is my talk.
Okay, so Juan Carlos, you completed the first part of your talk?
Sí.
Yes, perfect. So maybe we can have a little bit of discussion before the second part. And I have no words to say how many bells your talk ring in my head. And it's really full of inspiration and full of new, fresh idea, I think, even if the topic of autopoiesis is very classical now. But this idea of inference and the Bayesian probabilities, for me, is very interesting and maybe also possible to develop at a minimal level, even experimentally. So there are questions. Oh, yes, Luisa.
So, thank you very much for your talk. It's really amazing. I totally agree with Pasquale. I wanted to ask a question. So I try to ask a question. So if in the process of structural coupling, the autopoietic system is supposed to react with self-regulation processes to the perturbation. And this is defined as a cognition in sense of generation of meaning. That is the association of self-regulation, of the pattern of self-regulation to the external perturbation. So you are in the process of, this is my question, are you in this process of interpreting and describing this process of generation of meaning as a computation of Bayesian probability?
Yes, I reached the conclusion that internally we are intrinsically probabilistic machine that do Bayesian inference.
But this would imply that the generation of meaning is a sort of computation in a computationalist way, or are you anti-computationalist like Maturana?
I am anti-computation, no, because, but it's, no, myself, I'm not the word computation, but probability, you know, that you know.
Okay. No. I wanted only to be sure of this.
Probability.
So this generation of meaning is an anticipation of what could be, is a creative act. Is a creative act.
Wait, very important. You just mentioned the most important word that I forgot in my talk.
Okay.
These machines are, all of them are anticipatory systems.
Yes, this was the great idea of Maturana already in 1969.
And it was the great idea of Rosen.
Yes, yeah.
He wrote a book called Anticipatory System. And you can anticipate because they are a dynamical system with probabilities, and they are anticipatory systems.
Okay, so you are deepening this. What is creation of meaning in terms of self-regulation as an anticipation? Anticipation. Okay. This is absolutely interesting. So I would be immensely happy to know more about this, but maybe outside the context.
We should have it all because I have ideas about funding. Yeah.
And this is also very interesting. Thank you so much. Thank you. Really inspiring.
Okay, other questions? I have a question, Juan Carlos. I'm a little bit interested recently in how implementing the chemical domain, say, some computation, like neural networks, as mentioned before, but also Bayesian probabilities. I don't know exactly where to start. Maybe you know if someone tried already to make a chemical Bayesian machine. I don't know if the name is correct. Or a network, something that can work as a Bayesian network.
No, really, very interesting question. Myself, I'm still thinking in abstract, you know?
Yeah.
I am not thinking like in a Belousov-Sabotinsky type reaction. Yeah. But
Okay, yeah.
if you think a little, Belousov-Sabotinsky type reaction is in some way, because of his oscillations in time and internal oscillation, more or less is a prediction about itself. You see, the machine, the state of the machine is a prediction about its own future. It's a, I will go around this direction.
Yeah, it's something that must be developed. I was thinking to some, very simple stuff, but still no idea. I need to study more. Okay, if there are no other questions, we can move to the second talk, which is very, very interesting too, an historical travel. And we go in Chile in the 60s. Okay, perfect. Please.
I selected the same. Yeah, something is happening here, of course. Yeah, there.
Yes.
There, yeah?
Yes.
Then this is Cybernetics and Chile, and I am Juan Carlos Letelier from the University of Chile, and there you have the pictures of very important people. At the left, you have Humberto Maturana, and at the right, you have Heinz von Foester in a picture of the 1960s. Then let me explain you the life, because the life of Humberto Maturana, because it has very many interesting aspects. Maturana was born in Santiago, Chile in 1928. And he just died last year. He died of, he was 94, something like that. Yeah. He was born in 1928, and he contracted tuberculosis very early. When he was young, in 1940, he had tuberculosis. And he spent initially, the classic treatment for the time was to go to the mountain, do not eat anything. He went to the classic treatment, but also he received one of the first, he was one of the first recipients of the new wonder drugs called antibiotics. And then he cured himself of tuberculosis. And then he entered the School of Medicine at the University of Chile in 1950. But in the School of Medicine, he discovered that he didn't want to be a doctor. He wanted to be a researcher. And the School of Medicine, Chile in 1950, we didn't have science. Then he convinced the people to help him to go to England, because England, England, to go to England in order to be a famous anatomist, and he will return to Chile to teach fancy anatomy. And then he got a small fellowship, and he went to England to be under the guidance of a very, very, very distinguished fellow called J.Z. Young. This is very, very distinguished, amazingly famous biologist. And Maturana went to study with him. He arrived in London in 1954, but the British system didn't permit Maturana to study in England, because they said to him, look, who are you, Maturana? You come from Chile. Where is this country? No, you don't have degrees from institution that we recognize. Then no, go back. No, we don't want you to. You cannot do. You are not a doctor. You flunked out of medical school at 4 years to come here. No, you are a mess. Then they didn't accept him as a PhD student. But Young, he knew by then that Maturana was quite a personality. Young wrote a letter, and Maturana then traveled to Harvard, where they accepted him, and he did a PhD in Harvard. about the, the maturation, the myelization of the optic nerve. And he did the postdoc with a very, very strange and wonderful person, character, Jerome Levy, at MIT. And at MIT, they produced in 1959, 1960, a series of papers that were extremely famous in neuroscience called What the Frog's Eye Tells the Frog Brain. And these papers were published in the Journal of the Association of Electrical Engineers, not in biology papers. And there you have at the right, here at the left, you have Ledwin, at the center Maturana, and in the two sides you have McCullough and Pitts that were also mentioned here today, that they have produced in 1943 a paper about a theory how to build a computer or thinking machine using neurons. Then these four guys, these four friends, published this very, very influential paper about neuroscience. And then Maturana returned to Chile in 1961 and created the lab where I am still in this lab. I am in the lab created by Maturana in 1961, and this is one of the few labs in the world that is 60 years old. Labs, usually they don't last so long, but this has lasted a long time. Now, a little intermezzo. We are going to forget Maturana for a while. I'm going to introduce another character who is Hein von Foester, and nothing is better than to introduce the Macy conferences. When the Second World War finished, a philanthropist, a US philanthropist, decided that he will use the money to bring all the famous people that he can find to New York once a year to discuss the big scientific problem of our era. And these were called the Macy's Conference because the philanthropist was called Josiah Macy's. And the big conference happened between 45 and 54. You had a small conference before 45. And by example, in this, the people who went to this conference are a who is who of modern science and philosophy. They went von Neumann of computing, Norbert Wiener, Margaret Mead, Gregory Bateson, Claude Shannon, Hutchinson, the ecologist, J.Z. Young, our friend J.Z. Young, Gray Walter, a cybernetician, McCullough, Pitts. Here in this picture, by the way, here you have the central character, the person that is like the king of the meeting. The king of the meeting is Warren McCullough. And here in the side, you have a guy who is called Hein von Foester, who is a physicist from Austria, who arrived in the US in 1946. And Warren McCullough put him in charge of being the secretary of the proceeding secretaries of the Macy Conference in order to polish his English. Yeah, you're going to be how important is this is Hein von Foester. And by example, to show you the importance of this conference is the topics that they study. By example, in 1952, one of the topics was the relation of cybernetics at the micro level to biochemical and cellular process. You like the topic in 1952? It's like the topic of this workshop. Or in 1953, studies on the activity of the brain. You like? Both of them are very relevant to our workshop. Then these were the Macy conferences. But in 1957, a big event happened, and the big event was that the Soviet Union launched the Sputnik. And when they launched the Sputnik, the Americans couldn't believe that the Soviet Union has beat them to the technological reason. Then Uncle Sam was desperate. Then in 1958, they went into a frenzy of creation of things, of organization, in order to jump start. This is how they call it, to jump ahead of the Russian, to close the technological gap. Then in 1958, look, they created 4 things in the US. They created NASA, DARPA, the big Department of Financing of Exchange Project. They created a law, very interesting, the New Educational Act, that was the first law to produce free college education in the United States for people who wanted to study science, technology, and languages. This is quite a unique thing. And they created something that they themselves, they created the laboratory of the future that was called the Biological Computer Laboratory, BCL. And the director and unique director of BCL was Hein von Fester. And the goals of BCLs were quite ambitious, was to develop intelligent machines, a new method to program computer using the perceptron as the fundamental idea. And the funding came 100% from military sources, DARPA and the Navy. And by example, here you have, in 1960, the BCL, in order to appear in society, they organized like the ball, where the great ball, you know, where you present ladies in society. They organized a ball that was called First Conference of Self-Organization, produced in June 1960. And this is the Conference on Self-Organization. And this picture is taken at the, at the place in the University of Illinois. And it represents of who is who of system thinking. You can here find von Fester, von Bertalanffy. Von Bertalanffy is here, I don't know if it's there. Hayek, the great economist, I don't know if it's great, the famous economy, McCulloch, Ashby, Gordon Pask, Rosenblatt, who invented the perceptron, Sperry, the experiment of cutting the corpus callosum, Stafford Beer, that is going to be very important. And interestingly, in this group of people, you have three of them, von Fester, Beer, and Hayek, that will end playing important roles, but in opposite trenches in the politics of 1970s in Chile. Interestingly, no one from Rachesky's group was invited. Even the Rachesky group was in Chicago, 70 miles away, and they have just published the fundamental paper of MR systems about biological organization, and they didn't invite Rachesky or Robert Rosen. It's typical thing of the academic life. And here I point for you to know, this guy is Stafford Beer, here is Hayek, and here is von Fester. And these are going to be very important in what continue in the next part of the story. Then, now we return to our main character. In a neuroscience conference in Holland in 1964, Maturana met von Fester. Von Fester, who had a huge budget, traveling budget, he traveled all the conference in order to try, because he had to understand the brain in order to produce the new computers. He went to many neuroscience conferences, and there he met Maturana. And it was an instant friendship, to the point that von Fester visited Chile for 4 months in 1965. He spent 4 months in Chile, and he gave some seminars on cybernetics at the medical school and at the engineering school, and he stayed at the house of Maturana. And von Fester will always say that this period of his life, where he was, for the first time since the war, he was like away from the universe. He felt free in Chile, talking about science, and didn't have the pressure. He didn't have the internet, then nobody was bothering him 100 times a day. He spent 4 months in Chile, and Maturana reciprocated the visit, and he spent like 11 months at BCL lab in the period 68, 69. And in that period, Maturana wrote the fundamental text of all this story, and the fundamental text is called the technical report number 9. It's called Biology of Cognition, and there you don't have any concept of autopoiesis. There the only concept that you have is that the nervous system doesn't detect reality. The nervous system It's always in a loop, perception, action loop, and that's it. Yeah. And this is the fundamental text. And this is why you have the Ouroboros is the symbol of all this theory, because the action perception loop. And for you, if you don't know, in 1970, Salvador Allende, who was a socialist, was elected president of Chile. And the idea of having a centralized economy, who already was a very popular idea in Chile, took a lot of force. And I need to explain another detail. This is another intermezzo. In 1939, Chile suffered a big earthquake. And because of the big earthquake, half of the industry disappeared overnight in Chile. Then the new government, wow, what happened here? Excuse me, the system reset itself.
Okay. Yes, we can see again. Yeah.
Then the new government, who was a socialist government in 1939, was a socialist government. They decided to create a big state-run institution to run the economy. Like in Japan, it will be like a small BIT, you know, a small minister in trade and technology, you know. And Corfu was following the lead of something that has already been created in the United States called the Tennessee Valley Authority. That was a special entity that tried to push the economic development of the valley of the Tennessee River. Then Corfu began to take charge of more and more aspects of the Chilean economy, and by 1970, Corfu directed 25% of Chilean economy. Then Chile was not a socialist country, but it was a centralized economy, more or less well run. And this Corfu entity, that's the state run, directed 25% of the economy. And this was the golden age of the Universidad de Chile. Corfu expanded the Universidad de Chile a lot. Corfu imported mainframe computers. You know, Corfu was into the business of technological development. Now, this was in 1970. In 1969, Varela, who was a student of Maturana and was doing a PhD at Harvard, went and visited BCL2, and there another friendship happened between Varela and von Feste forever. And in 1970, Varela returns to Chile. Maturana was already in Chile after the sabbatical year at BCL. And in 1971, von Feste spent a complete month in Chile teaching cybernetics at the Universidad de Chile. And something else happened in 1971 is that a Corfu top brass, a Corfu chief called Fernando Flores, decided that Chile will have a scientific economy based on cybernetic principles. And this project was the Project Cinco. Yeah? And then the Chile will have a scientific socialist economy. Yeah? This is, you know, people began totally crazy stuff. Yeah. And also in 1972, this book, this red book called De Máquinas y Seres Vivos, was printed in Chile. And it is in this book for the first time that the word autopoiesis is written. This is the book where appeared the definition of autopoietic system in Spanish. You need to wait a long time until 1980 to have it in English. Yeah? And in 1972, the great British cybernetician Stafford Beer accepted the offer from the Chilean government in order to come to Chile and be the boss and the big boss of the Cinco project. And Stafford Beer went to Chile, came to Chile, and he took the job, but with gusto. He bought a house. He found friends. He decided to be a Chilean, another Chilean, and he was the boss of the Cinco project in 1972. Yeah, and here you have, in the top, here you have, this was the control room. This is amazing. It's amazing. Huh? This is the control room of the Cinco project. Yeah. And the idea that in this room, people will gather in this fancy desk with have some of type of with keyboards, and they will decide that Santiago needs tomorrow 2,000 lettuce and 400 liters of milk, and they will command the production of lettuce and milk, and the production, the repartition of goods and money will be the most efficient things that you can imagine. Yeah. And here on the top, you have a picture of a Stafford bear, the guy with the, with the bear, Stafford bear, and Fernando Flores in Chile in 1973. Yeah. Well, they were working 20 hours a day in order to produce the famous Proyecto 5. And in 1973, von Foester stayed another month in Chile, in Santiago, and he taught a course called The Cybernetics of Cognitive Processes, and the course was aimed to the technical staff of the 5 project. And this course was the best course on cybernetics ever taught in the galaxy. And we have the course because some guy, one of the helpers of him from Foester, came with him from the US, and he took copious notes, and we have now this document where it's amazing. It's the best course on cybernetics. And this was in July 73. And the teachers were von Foester, Maturana, Varela, Beer, Wilson, Schemenberg, who was a German guy interested in cybernetics. Unfortunately, in September 73, we have a military coup d'etat, and everything finished. The Project 5 was killed overnight. Varela escaped to Costa Rica. Flores, the guy in Corfu, was sent to jail. The Stafford Beer, who was at that moment of the coup was in England, didn't return to Chile. Maturana did continue in Chile. And von Foester extracted many people from Chile, saved them, especially Henry Uribe, who is the third author of the paper of autopoiesis. The paper where they describe the protobio, this cellular automata, has 3 other authors, Varela, Maturana, and Uribe, who programmed the computer. Then von Foester took Uribe to the United States and hired him at BCL. And Uribe was the last person hired at BCL because in 1975, the funding agencies of BCL cut the funding and BCL disappeared. The real reason is because BCL didn't produce the gadget that the Navy or DARPA wanted. This was the real reason. Then in 1975, Maturana returns and spent 3 months at BCL. He spent there just before the closing of the lab. And when the lab closes, von Foester retires from the University of Illinois, and he will be retired for the rest of his life. Then in 1980, the book Autopoiesis and Cognition was printed in English, and this was the first time where people in the English world could read about all these ideas, And essentially this book is 2 books in one, is the technical report number 9, this is the part of cognition, plus the red book on autopoiesis, printed in Spanish. This book is the 2 books put together. Also, I must not forget that in 1975, the other guy in the picture, von Hayek, and his friends, mostly from the University of Chicago, they take control of the Chilean economy, and the Chilean economy became the poster boy of neoliberal policies, and Chile became a producer of commodities, and we have to forget fancy stuff like zinc and cybernetics. Yeah. In 1993, Maturana Varela produced this book, which is called The Tree of Knowledge, that is the book where they present the core ideas of the theory to the general public. And amazingly, this is hard to believe, but this is true. Amazingly, this book is the outcome of a semester-long seminar that they gave to the generals in the Pinochet regime. The generals in the Pinochet regimes were very, 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 very interested in systemic thinking. They wanted Chile to be a powerful country. They wanted to have to produce a steel railroad, rockets to the moon. They were very, very interested in this. Then Maturana and Varela gave, imagine, it's amazing, it's really a long, one semester long course to the generals about autopoiesis and biology of cognition. Yeah. And bueno, and the other things, the autopoietic ideas were presented in Artificial Life #1 at Los Alamos. I was there, and no one was impressed, with the exception of Kern Smith, who is this scientist who claimed that life began in the interface with clays. And I couldn't give my talk. You know, they didn't say, no, your talk is too philosophical. No, no, this is, you know, no, it's true. You're coming from New York and he's Los Alamos. I couldn't give my talk in the first Artificial Life conference. Yeah, and I want here to see that we are here today because of this very deep friendship between Maturana and von Foerster, with a friendship with a very long consequences, lasting consequences. Without this friendship, nothing would have been possible. That's it. Thank you very much.
Thank you. Thank you very much, Juan Carlos. This is a very fascinating story, and tell us how politics and science mix together, and the personal histories of people, personal reciprocal sympathies. Of course, they are determinant to develop even scientific ideas, right?
Mm.
Okay, so it's we have time for some questions. If someone would like to ask curiosities about this. Okay, maybe Luisa.
As usual, I would have billions of questions, but I would like to ask what you think is the influence of Heinz von Foerster theoretical production on the theory of autopoiesis, because he was there, and he was there with all a theory of self-organizing systems that inherited a lot also from other traditions. So I wanted to ask you, because I know that Umberto Maturana always denied any genealogical relationship between the theories of self-organization and autopoiesis, while Varela was much more open, especially towards Jean Piaget, towards also Heinz von Foerster. So I wanted to ask you what you think.
No, I am sure that he was much influenced. When you read the Spanish book, you know, or the book De Máquina y Seres Vivos, where they introduced the idea of autopoietic system, this is a book from a cybernetic. The first pages are from the cybernetic ideas. And without any doubt, Maturana learned these concepts in his sabbatical year BCL, where Gordon Pask, who was a very famous cybernetician, was working. Yeah. Then there you have a clear influence. And
also all is coming also, as I was mentioning before, in my view, from still McCulloch and Pitts, because there is always this.
Well, claro. And Maturana has worked with McCulloch and Pitts, you know, and Maturana was very close with Jerome Levin, who maintained, you know, Jerome Levin, Pitts lived all his life with Levin.
Yes, yes, yeah. Yes.
Then there you have a very intimate mélange, I will say in French. Yeah.
So you may agree about with the idea that the organizational network that they are drawing in the theory of autopoiesis as the organization of the autopoietic system could be an evolution of the idea of network that was in McCulloch and Pitts, not maybe in the 1943, but in the 1947 article, where there was already an opening towards autonomy. And then this idea evolved in Heinz von Foerster, and then evolved also with Maturana and Varela. Would you see a line?
Without any doubt. And the contribution Maturana, this doesn't diminish. But Maturana was always, he would say, no, 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 no. But without any doubt, because they talk about a machine. And the only people who talked about machines were the cyberneticians.
Thank you so much.
Okay, very good. There is a comment in the chat by Seth Bullock. Maybe if Seth would like to even talk, it's okay.
I know the story why Varela was the editor of the Artificial Life Europe.
Yeah, sure. So I just wanted to point out that Maturana and Varela's ideas were welcome in at least some parts of ALife. Initially, ALife had an American conference series that came out of Santa Fe and a European conference series that came out of France and a lot of European philosophy, and they had quite a different flavor. They really felt like different, but different perspectives.
And I know the stories because for the second Artificial Life conference, Varela sent the paper and they say, no, we are not interested in you. Yeah. And
so do you think that was driven by do you think that was driven by intellectual reasons, or do you think there's some politics in that, in the relationship?
No,
between...
intellectual. Look, I went to the first one. Yeah. And the first one, the only thing that they talked was about computers. Yeah. Computer was the model of the living system. Computers. Yeah. And but someone did talk about autopoiesis, but it was a very strange talk. Someone called Milan Seleni.
Oh.
But Milan Seleni, I couldn't talk with him. Milan Seleni, very strange what happened with him. But the first Artificial Life one was computer and program. Nothing else. Then Varela didn't believe in that. Then this is why he said, tough luck, I'm going to have my own Artificial Life. And he organized with Pierre Bourguin, the Artificial Life Europe.
Great.
Oh, quite interesting story. Yeah. So the European conferences are, let's say, come from, as a side direction with respect to the US Artificial Life. But now we merge again. We have only one Artificial Life community. Okay, great. If some other question or comment, I will remember that 1 year ago in the past Artificial Life, we also had a similar workshop to commemorate Umberto Maturana, who disappeared in May 2021. And so this time also we would like to commemorate him again. And if apparently there are no other questions or comments. Okay.
I have maybe another thing, if there isn't anything. Do you know or have a perspective on Flores's influence on AI in the US? So he obviously he went on to work with Terry Winograd, yeah, on who was really famously responsible for Shrudlu, which was a AI reasoning robot, and kind of influenced a more traditional AI branch. So do you see, I don't know so much about the relationship between Flores and Maturana and Varela. So I don't know whether you have anything to comment about.
No, I have also, no, I have, you must know that Flores hired Stafford Beer after he met in 1971 von Foester in the visit that von Foester did to Chile in 71. You know, Maturana took von Foester to Flores. Chile is a small country. And then Flores said, look, you know, Chile, we're going to cybernetize the Chilean economy. Who do you recommend, Dr. von Foester? And von Foester said, I know the character, Stafford Beer. This is the connection. Yeah? Then after the coup d'etat, Flores was in jail for 1.5 years. And Maturana went twice a week to jail to discuss the book about autopoiesis, you know, the Máquina y Seres Vivos, in jail with Flores. Then Flores appreciated, because of that, because it was a very difficult time, Flores did appreciate a lot Maturana, because Maturana went to jail, to see him in jail every week. And this
it's amazing
is why
to imagine what would have happened if the coup had not happened. You know, what the development would have been there without disrupting that coming together of such amazing people. But yeah, we'll never know. Anyway, thank you very much for your talk.
Yeah, thank you.
Yes, thank you. And we have a question from Chris. From Chris Gordon-Smith. Please activate your microphone.
You're quite right. I'm still doing that. Fantastic talk. Thank you very much. I mean, just a quick thought. I read, I think, most of the 1980 book last year, but I did find it, even though there's no mathematics in it, I did find it very hard work. And I'm wondering, could that be part of the reason why, you know, it wasn't accepted, the ideas were not accepted at ALife 1?
Without any doubt, it's a very dense book, no mathematics, but it's very dense. It's like written by mathematics that don't use symbols. It's true. And this recursivity is very recursive, the text. Yeah. And the first part of the part of cognition, that is BCL report number 9, it goes against the grain of the computer metaphor, because they said clearly the brain is not a computer, it doesn't detect reality. Imagine, then of course in the 80s it was impossible that these ideas were accepted.
Yeah. Thanks.
Yeah, and I would add, Juan Carlos, you know, to connect with the question of Chris. It was not autopoiesis, as Chris said, was not accepted in at the beginning in artificial life community. But I would say that even now, autopoiesis is totally ignored by biology community. So very often we speak with Pierluigi Luisi about why, and he said that the reason is that the biology is DNA centric.
See, it's true.
Oh, yes.
Also, it's because biologists are very concrete people. Biologists will accept anything if it is useful for them. It's because up to today, the theory is still primitive. We live like in the pre-Bohr, we live in 1912 in quantum mechanics. We have a very primitive theory where we cannot explain like the stability of metabolic networks or some interesting things about metabolism. But I have the solution. Yeah, then if anyone who wants to talk with me and we have the solution, we build the collective solution. Yeah, we need a technical result. You understand? We need a technical result that will convince people, not because it's philosophically perfect, but because it's empirically and heuristically potent.
This is a SBI mission, Pasquale.
And I know, by the way, I know which is the technical result.
Okay, perfect. Yeah, and with these wishes, we can conclude our workshop. I would like to thank everyone, of course, the co-organizer, Luisa and Yutetsu, and Silvia, again, appeared in the screen. So I thank you, everyone. Juan Carlos, thank you really much for your talk, and to all speakers.
Ash, thank you very much.
Thank you for inviting me.
Okay, so